All right, guys, does anybody have any questions before we get started with the lecture part? We're missing a lot of people today, but whatever, that's not my problem, right? I've had about like 6 people apparently are sick today, and how much I believe it, I'm not sure, but what can I do about it? I don't really care. It's not my points getting lost. So, you know, everybody feel better, I guess, if you guys know anybody that's sick, everybody feel better. So I get it. Like, we don't have a lot going on in lab, but yeah. So even, and I will say this too, like, just so you guys know, I know you guys are the ones here, but even on these days where we're not doing a lot in lab, like today, we're just going over what the lab project is, and I'm going to go over the lab exam also, so we can go see what, we missed and everything like that. It's still required attendance, and if it's a required attendance, even though we don't have a grade associated with, we don't have a pre-lab or anything this time, yeah, and that's just how it is. But even if you miss that day, it still counts as one of your lab absences. Remember, you can only have up to 6. Once you hit 6, you're out. So remember that. So yeah, just want to bring that up in case, like, because tomorrow, I know today is like the easy day. The next lab, say tomorrow, but the next lab for you guys, it's just going to be gram stains. So like, we kind of have to do those. So it's not going to be something we're going to get away with. And when I say we have to do those, basically like the tubes, how I gave you guys the tubes in the beginning, and I'm like, do the gram stain and figure out, which one's positive and which one's negative. I'm giving you the same tubes. I'm labeling them completely differently, and you need to figure out which one is your positive and your negative, and I will not be helping you this time. So you should know how to do it. I feel like at this point, you should not only know the steps. I'm going to give you the steps. No worries there. That part, don't worry. the steps of what stains even to use, even though we should know that. But the doing it, the acting it out part, what if you guys are having a problem finding out in your result, looking in your microscope, which one is positive and which one is negative after doing the gram stain, then I mean, I'm going to make you do it no more than 3 times. But if you can't get it after 3 times, then we've got some sort of problem there. So. But yeah, so that's the idea. I know at least one group in lab will have the freaking one that always stains positive, even though it is negative. If you are that group, then I will tell you that. No worries. Like, you already know that probably already, so you know what to expect. But no worries there. I'll help you out if you are that group. If you're doing your gram stains and you're like, I don't know, this is the positive, this is the negative. Yeah, you can come check with me. It's fine. That part will be fine. Okay. Do it, be like, okay, this is what we got for positive, this is what we got for negative. I'll be like, yep, you guys got it. Like, that's great. But if you guys are just not getting it and you can't figure out and you don't feel confident in it, or you think you get it and you're not really sure, but you don't want to come ask me if it is correct or not, then you will be doing every single experiment thereafter with potentially the wrong samples. So that's on y'all though, after that. So that's setting you up for the unknown. We'll go over it more whenever we get into that. But yeah, so this is just kind of reprising the gram stain with that, making sure that you guys actually really did retain the knowledge of how to perform the stain, even if given the steps. So yeah, when we get into lab, we'll kind of go over, because we're going to go over the questions, I'm also going to cover some of the theory so that you guys know why the answer is the answer, okay? Because that stuff you will see again on the final because it's comprehensive, so FYI. All right, so this chapter is chapter 15. It deals with innate immunity. In your immune system, well, in your body in general, you have essentially 2 ways that your body can fight off intruders: the innate system and the acquired or specific system. And the innate system is exactly kind of what it sounds like. It's an innate, built-in, general fighting concept kind of system. Think of the innate immune system as being like the frontline guys in the war. Okay, these are the Marines on the ground. I always think of like D-Day or something like that. Like I always think of like World War II when I think about my examples. But yeah, the guys that are on the ground actually like there, but they're just like trying to hold things together until the tanks arrive, until, the bomber planes arrive, or until the specialists arrive. So they're just holding down the fort, holding down the line, doing what they can to fight the good fight, but they're not going to be, the aces of the story, right? The aces of the story, that's going to be your acquired immune system. The problem is the acquired immune system takes time to build up. It is like going to a war, seeing that, okay, wow, we're going to have to fight this big fight and calling in for tank support or bomber support, but now you have to wait for them to get there. So what do you do in the meantime? That's the innate system, okay? So the guys that see it first, the guys that react to it first, and now we're waiting on the heavy, artillery to show up, basically. So we can divide innate immunity into two parts. What I feel like is not obvious immune stuff, the first line of defense, things like your skin or the salt on your skin or the mucus in your nose and sinuses and in your lungs, the acid in your stomach. All these things are still protective for you to keep out invaders, but they're not what we would think of as common, immune reactions, right? When we get into talking about the cells of the immune system, the phagocytes that we've been bringing up over and over and over again, the white cells that eat up the bad guys, right? That's going to be second line of defense. So I'm already telling you, like, oh, we've got all these phagocytes that eat up all these bad guys. And you hear how important they are in our immune system roles, because I just literally just keep talking about it all the time. And they're not even the artillery. They're not the big guys, not by far. So this is it gets pretty cool. I think it gets pretty cool understanding how all of this works. Now, whenever you guys hear, when you're doing your stuff in physiology, or when you're doing your stuff in nursing school and you're hearing about running a CBC and you have the white blood cell count and what eosinophils are and neutrophils are and lymphocytes are and what they actually do, as opposed to just being numbers that have to be within a range. So it's important I feel like, to understand that stuff. Because why would somebody's, lymphocyte count be so high? Well, if it's an infection, but why are lymphocytes high in an infection? So hopefully you learn that throughout chapters 15 and 16. That's what the whole point of these chapters is, right? So our second line of defense, these are going to be our basic frontline fighters. They fight bad guys. They don't have a specific attack goal, right? Like our antibodies, we've talked about how they can be very specific and how they interact with things. That's part of this system, the acquired, the third line of defense, okay? So they don't have anything specific like that. They just see, oh, this is bad guy. And yeah, they have things they'll interact with that signal them for that, but like they're not going to need to adapt for that specific thing or have a specific clone cell that they have to interact, like what we're waiting for over here. So that's useful though, because they're just ready to go. We don't have to wait on them. So those are our phagocytes. So we have phagocytosis. Let me use my pen so that the recording makes sense to people. Otherwise, they don't know what I'm talking about. With how much I write on the board and stuff, it's amazing anybody understands my lectures when I record them. Okay. All right. Phagocytosis. There's our phagocytes that are going to eat up bad guys. We already learned about a little bit about that and the importance of it. Inflammation, yeah, it has a role, as does a fever. These things are pretty closely related to one another, okay? And we'll talk about the role of what they are and why we have it and why it's important to have them as we go through this. And then antimicrobial products, things produced by cells in the area that help with dealing with invaders, okay? These are just general response things. We all know that you can have inflammation in response to any kind of cellular damage. It doesn't have to be an invader, right? You can go stub your toe and get inflammation from that. Literally on a Sunday, my husband dropped my kayak on my toe, and my toes, I guess, all of them. But they're not light, They're not light. And so it really hurt. I knew they weren't broken or anything, but inflammation, it turns red, it gets swollen, and it's going to bruise and everything like that. But there was no invader really there. I didn't have any open wounds or anything. So that's just an example of how inflammation can play a part in cellular response when it doesn't have to be always in fighting an invader. Same with phagocytosis. The cells that are going around eating the bad guys are also going around eating cellular debris. So dead cells that just die naturally. We know they don't live forever, or even cells that were damaged from some sort of injury. They'll go up and take care of that debris and break that down too. So they play a pretty wide role. They're always there. They're always doing stuff, and their roles are always important. You probably right now have some sort of inflammation going on in your body. Almost everybody does at any given time. You can probably tell me, oh yeah, my tooth hurts. Or oh yeah, my toe is sore from the kayak that got dropped on it, or whatever it is. We often have a lot of this stuff going on already in our bodies. We just don't think about it. So immunology basically is talking about the surveillance of the whole body for foreign invaders. So it's nothing crazy there. We kind of learned a little bit about that. So we need to be able to recognize what is foreign versus what is self. How does our body know the difference? And it's easy enough to say we are trained to recognize self and non-self by recognizing surface markers. Now, this is the answer. And if I was to ask you a question about how we tell the difference between self and non-self, yeah, it's by recognizing surface markers. But man, that's a pretty vague statement. There are all sorts of proteins on the surface of your cells that do all sorts of things. Either they are interacting with their environment to receive chemical signals from nearby, or they're interacting with invaders themselves, or they're interacting with other nutrients in the area, or they're channels that are allowing things to pass in and out of the cell. And all of these things that are going on in the membranes of your cells right now, those things can be identified by your body as being self or non-self, right? So hopefully those things, if that's part of your body would be seeing that as self, yeah? So it trains itself as that. What it'll do is if it has any cells that react to something that is also recognized as self during the maturation of these cells, then we literally kill those cells. We don't let them multiply. There can be problems with this whole scenario because if they don't get deleted properly, like let's say that we're activating things related to it nearby even, and somehow that checkpoint system doesn't catch the self, and we accidentally allow ourselves to see it as non-self, then you can develop things like autoimmune system, disorder. So things like if you have an autoimmune disease like lupus and rheumatoid arthritis, all of those is what I'm talking about here. So that's the same immune system that we're dealing with. It's all the same. It's just what it's reacting to, reacting appropriately to, E. coli, or reacting inappropriately, seeing your own, endothelial cells of your, blood vessels as foreign, for example. Another way we can look at this is like the markers on the surface of cells. We see them as self, and it's so specific that if I take my kidney and pop it into your body, it's likely that you'll reject it, right? What does rejection mean? Your immune system is killing it because it sees it as foreign. That's what that is, okay? That's why people who get transplants get tested rigorously to see if they are going to match genetically, and we try to get them as close as we possibly can. We run tests to make sure that the antibodies that the recipient has already in their body aren't going to react with their donor tissue, and we'll have even their lymphocytes in there and see if there's any interactions with that, and we'll do testing based on that. And do this if you're somebody waiting for a kidney, we've got all of your serum from like years of you waiting for it that we've been testing to see what antibodies you have in your serum, just to see which ones you have, so that we can try to find when a donor pops up, this guy, he meets that profile. He doesn't have these things that they have antibodies to. So this should be safe. Then we go ahead and we match them up in this elaborate friggin', immune battle essentially, and see if there is any outcome of reaction there. And if it doesn't react in that system, well, that's happening the day that organ came in. That's happening the day that patient is laying on the table, ready to go, waiting for the surgeon to give the go ahead for that. So they're probably waiting literally in that prep area where you do for surgery, waiting to hear the go ahead of, yes, you're going to be a good match with this kidney, and transplant immunology gives you the go ahead. It's like the most stressful job in the whole world. I felt like it was the most stressful job in the whole world. That's what I did, that testing for Integris, and Integris Baptist is really the only hospital that effectively does this in the Oklahoma City area. OU does a little bit of it, but they're not anywhere near as effective at it as Baptist is. So if you guys are looking into transplant medicine, Baptist is where you should be going. In fact, all of the scientists from OU's lab quit and moved to Baptist. So it's like working with all OU people for a little while. But yeah, so that's what we would do. And so it's so specific. There's all these different markers that you could interact with and see as foreign on somebody else if they have them. And their version of those markers might not be the same as somebody else's. So we do literal genetic testing as well to see how closely their genetics might match up with yours. It's super specific. It's very complicated stuff, but human and human isn't self, you know, like that's what my point is here. There's so many different markers on your cells that you would see my cells as foreign because my markers are just different from your markers. Do we have the same kind of markers? Probably, but yours are just genetically different enough from mine that your body would attack them. It's pretty interesting and also kind of, I don't know, I don't want to say terrifying. If you need an organ, it's terrifying. But yeah, my tattoo artist, she had a lung transplant. And I thought that was just kind of crazy that I was, you know, someone that worked on that stuff. And she's had a whole like double lung transplant. And she's fine now. She like lives a pretty normal life, you know, other than she has to take medication for that sort of stuff. But you always have to have immune suppressants after that. But yeah, okay, moving on. Anyways, antigens. We've learned a little bit about antigens and what antigens are. As I'm sitting here talking to you guys about these surface markers and how yours look different and we react foreign, that would make those antigens, right? Your body has the ability to react against them and see them as foreign. That's an antigen. That really is all an antigen is. It's nothing fancy there. So what are antigens typically made of? We'll learn way more about this in chapter 16, but just briefly, they're typically going to be proteins and or sugars of some kind, usually protein with sugars on it. You know how I've mentioned in the past glycoproteins? That's what we're talking about. So those markers on the surface of cells tend to be glycoproteins, FYI. Anything that can be seen by the immune system, though, and reacted to can be an antigen. All right, so so we have to be able to see what is foreign and be able to react to that appropriately. And anything that is self, be able to tell our body, we do not react to this. Your gut biome actually has quite a lot to do with this because you have to get used to those bacteria that live in your gut and not attack them all the time. So they train your immune system on when to react and when not to react as you're developing through age. So they're very, very important in that sort of stuff. Anyways, autoimmune disorders is whenever that whole signaling of self versus non-self gets messed up somehow. We can even have bacteria like strep. Strep itself is, it has markers on its surface that look a lot like our surface markers. They're different, but they look close enough that if your body reacts to it just right, it'll also react to your own markers on your surface of your cells because it'll get that stimulation from saying, oh, this is bad guy. And then, yeah, and then that's a problem, right? So that's how things like autoimmune disorders can happen, if not only just from being, you know, genetically related and all sorts of stuff too. We'll get into that in chapter 17. Yes, 17. Okay, so how are these cells seeing self versus foreign? These innate immune cells don't see things specifically, right? So they're going to see this as, is this foreign or is this not foreign? Is this bad or not? Like it's very basic. So what they use, they recognize pathogen-associated molecular patterns, PAMPs. It will be on the exam. You'll have to recognize the choices. I'll say, what does PAMP stand for? And then there will be several choices and you'll have to know that it's pathogen-associated molecular patterns. Yes, they will all be PAMP words. So be ready, because I mean, obviously, if it's not PAMP, then I'm asking the question. So it's going to be that. So be able to recognize what those words are. Hopefully, as we go through this, it'll make sense that those words make sense in this context anyways. Pathogen-associated molecular patterns. So this isn't anything crazy that, this isn't anything crazy that we're trying to bring up something new that we haven't ever seen before, something like this. What is going to qualify as a pathogen-associated molecular pattern? Something that we can always rely on as we can recognize as bad guy. There's three things, and it will bring them up later too, but three things that we can name right off the bat. Peptidoglycan, right? Our cells shouldn't have that. That shouldn't be anywhere in your body other than in your gut area where we've already learned how to deal with those microbes, right? So having seen peptidoglycan, especially in your bloodstream, or outside of where they're supposed to be, bad, instantly, right? So cell wall bacteria, bad. The other one, LPS, which AKA, that's lipopolysaccharide, AKA endotoxin, which is found on the surface of gram negatives, right? So all gram negatives have that on their outer membrane. So if our body sees that hanging around, immediately bad guy, right? Another one that we see as immediately bad guy is not bacterially related, is going to be double-stranded RNA. So most of our body doesn't have proper double-stranded RNA, right? Most of it is going to be mRNA, and then you have the tRNA folds up in the weird T, but it's not true double-stranded of 2 strands, right? Viruses can have that as part of their genome. So if we see double-stranded DNA hanging out in our bloodstream, our body assumes, bad guy, this is virus immediately. So those and there's other ones, but those are ones we've learned about and we have a concept so we can understand them. Are PAMPs. They're pathogen associated molecular patterns, right? So they're things that you can immediately associate with that pathogen, that it is a pathogen. You don't even have to narrow it down to E. coli or anything like that. You know, this shouldn't be here, pathogen. That's what these are, okay? They're recognized by something called pattern recognition receptors. We're gonna maybe bring them up a little bit later. I'm not gonna test you over them, but it just happens to be the receptors that recognize PAMPs, okay? It's a general term. There's a whole bunch of them. All right, so PAMPs are important. That's what we recognize as far as kicking the innate response into gear and actually starting that response, right? Hey, this is a pathogen. This is a bad guy. We need to go to fight. They don't care about exact what it is. So anyways, next we have the breakdown of things that are important in the immune system. And we'll come back to some of these, including the mononuclear phagocyte system. It's also not as fancy as it sounds, but then we have extracellular fluid. This is literally just fluid around tissue, within tissue, between the cells, anything filling in the spaces, okay? And in those spaces are called the interstitium. So those are the extracellular spaces filled with fluid. And then we have the bloodstream, obviously. So all this stuff often is getting transported around in the bloodstream, as well as the lymphatic system, okay? It is separate from your bloodstream. It has its own little vessels that it'll go through, but it's completely different how it moves throughout your body compared to how your blood does. All right. But anyways, let's go. Let's do. So the whole point of the lymphatic system, which we're going to, obviously these 2 chapters are going to have quite a lot to do with the lymphatic system. So your lymph nodes, the lymph itself, the vessels of the lymphatic system, all of these things, and the organs involved in it, involved in your immune system. Okay. So it's just the whole system is meant as a route for fluid in the tissues to get back into the lymphatic system, where we're going to filter it out, start interacting with things with our identifier cells and things like that, as well as just rejoining that fluid back up with the bloodstream. And we'll talk about how that works in a moment, but it's kind of like it says, a drain off for the inflammatory response. You, let's say, stub your toe, okay? And we've already said that it's going to swell up and turn red and get, you know, hot and all this stuff. Well, fluid, it's swelling because fluid is rushing to that area. It's not just swelling magically. How does the swelling happen? It's fluid from your bloodstream rushing into the area. Why is fluid rushing to the area? Because chemical signals are moving into the area, as well as mostly cells that are responding to whatever that inflammation is. Because your body's already sent out chemical signals of its own saying, inflammation, 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 we need it now. Like, we need to start doing this. And so all that fluid rushes in there with all the cells that are going to come fight the good fight, right? That has to get out somehow. So when it leaves the area, that fluid, when it moves back out, that's draining off into the lymphatic system. Okay, so that's lymph at that point. That's what lymph is. Okay, it's the fluid of the inflammatory response, if you will. But yeah, so that whole point of the lymphatic system is for like a highway for your immune system, essentially. So yes, you're draining off all that fluid, but yeah, it's not all it's doing, right? It's also with that fluid bringing all of those cells with it, and those cells and antigens and everything with that going into the lymphatic system, where we're going to run through some lymph nodes and all sorts of stuff and react how we need to react in response to whatever thing we brought with us. So the lymph itself is like plasma-like fluid. It's formed whenever we have blood components from the blood vessels into the extracellular spaces, and then into the lymphatic capillaries, especially when we're bringing it back. That's when it is truly lymph, okay? I'll tell you this, well, we're gonna get to them. I don't wanna get too complicated with it now. Okay, so the lymphatic vessels themselves. They're very thin walls, the lymph vessels are, because we have to keep having this transfer of fluid and cells across, right? So we have very thin walls, and cells can squeeze in between the cells of the vessels and all sorts of stuff, because it's made for that, right? For moving into the tissues and out of the tissues. So we have to be able to do that. Okay, differences between the bloodstream and the lymphatic system. Okay, so I'd be aware of these. Lymph moves in one direction only, from the extremities back to the heart. Okay, how did it get there? How did the fluids get there then if it only goes back? Well, it's fluid from your bloodstream that went there originally. And that fluid from the bloodstream now is coming back through the lymphatic system as lymph fluid. Okay, so it came from the bloodstream. It wasn't technically truly lymph, right? It's just bloodstream fluids. And that's how it got there in the first place. Now we're coming back as the lymph, coming back and moving back to the heart to get circulated back into the bloodstream. It's important, though, that we go through the lymphatic system and not just back into the bloodstream, because we have to get filtered through things like lymph nodes and stuff like that so we can interact with our immune system properly, right? And get responses against foreign things, right? Lymph, so it only moves in one direction, from the extremities to the heart. Lymph is only transported through the contraction of skeletal muscles. Another thing that people often don't realize, if you have worked in a hospital or had a surgery or had a loved one who has had surgery, and after the surgery, very shortly after, as soon as you seem like you are all like with it, they'll be like, you need to get up and walk around, right? They'll get you up, moving around ASAP. If you can't do it, they'll put those things on your legs that squeeze your legs alternately to move that. That ain't because your blood, honey, that's because your lymph. Okay, it does not pump itself. So if you don't move around, your lymph won't move back to your heart. So then what happens? Edema, right? So you get swelling in areas, especially in the extremities, because it's not able to rejoin the bloodstream. So it's just moving out for inflammation reaction and then just staying there because there's no way for it to come back. You have to move to get the lymph flowing. It will not flow on its own. Absolutely will not. The bloodstream, the heart will not pump it. It's not how it works. So that's why they want people to get up and move around. It's not that they want you to get exercise and burn calories or something or build muscle. What they want is for you to move your lymph around so we can get your white blood cells going. We can start healing better. So you're less likely to get an infection. The sooner you move after a surgery or something like that, within reason, the more likely you are to heal without any consequences or anything like that, infections especially, because you want that lymph to move. So it's only transported through the contraction of skeletal muscles, right? Moving around, walking, moving your arms and whatever. If you are even confined to a bed and you're just not feeling well and you don't want to walk around, at least like move your arms around or something to help move the fluid. But I do recommend, if you aren't feeling well and you have kids that aren't feeling well and you are capable of walking around and you don't have like 105-degree fever or something ridiculous, you know, if you're average sick, then try to get up and walk around every hour. You know, just walk around the house, maybe make a lap and then go back and lay down. So you're way more likely to recover faster if you do something like that. So a lot of our lymphatic system will overlap with our circulatory, but clearly it is not perfect. We have quite a lot of just like these little guys coming off as vessels off into the tissues and everything like that, because just how the lymph is going to operate works differently than the circulatory system, right? It's different. We also have these little areas on the body where we have clusters of lymph nodes, right? And we'll talk about lymph nodes and what they are and how they work in a moment. But those little clusters of lymph nodes, you can reach into like your armpit sometimes and feel your lymph nodes in there if you have some sort of infection or something going on there, or everybody knows you can feel them in your neck. Yeah, especially if you have like a flu or something like that and they'll swell up really big. Or in your groin. Those most people know about these areas where you have lymph nodes. You have lymph nodes kind of all over the place, but those are obvious collections of the lymph nodes where they're going to be. And then we have lymphatic tissues that don't have true lymph nodes, but they have tissue that works the same way as a lymph node. It's just not in that same structure. All right. This is just talking about that stuff. Okay, so here are some organs associated with the lymphatic system. The primary lymphatic organs, the ones that are the most, like what we would say most important for generating your lymphatic system, the bone marrow. This is where all of your white blood cells and your red blood cells, by the way, come from. So they originate from this. That's what your red bone marrow does. That's what it exists for. It is literally for making your blood cells. And you might be like, oh yeah, but they deal with the bone marrow whenever people have like leukemia. Yeah, that's white blood cells. So let's just say that's white blood cells, right? So we're going to get into all of that. But a leukocyte is a white blood cell. It's the same word. Okay. It's just a different way of talking about it. And a lymphocyte is a type of leukocyte. Okay. But anyways, they all originate from the red bone marrow. And then they go to wherever they need to go mature and whatever. T cells, which we're going to talk about when we talk about lymphocytes in the third line of defense in the next chapter, they all mature in the thymus. And T cells are the only things that mature in the thymus. Okay, that's all your thymus is for. You might, if you've taken anatomy, maybe you've learned about the thymus and where it is. When we look at adults, especially the ones that we tend to get for anatomy, we have some younger ones. I think that the girl was like 38 or something like that this year. But one of the females, I don't know anything about it. But anyways, I heard that. And basically, your thymus is going to shrink down and be almost non-existent by the time you're an adult. So somewhere around like 25 to 30 years old, you like basically don't have it anymore. So most of the developing of your T cells is going to happen when you're a kid. And that's why it's so important to get kids vaccinated, not just to protect them while they're young, you know, and protect them for their life, but because their immune systems are developing then, and it's better to get them exposed to these things while their immune system is at its strongest and most able to build. Okay. All right. So anyways, that's our primary lymphatic organs. That's where everything is going to come from. Then we have our secondary lymphatic organs. This is going to be our lymph nodes, our spleen, and salt, malt, and galt is going to be like associated lymphoid tissue. Okay, that's what the ALT stands for. Okay, so A is associated, and we're going to talk about it, of course. Lymphoid tissue. So that's the ALT, right? So what's the S, the M, and the G? S is just skin-associated lymphoid tissue. M, mucosa-associated lymphoid tissue. G, gut-associated lymphoid tissue. It's nothing crazy. We're just talking about lympho-type tissue that's like what we see in lymph nodes, but it's in diffuse tissue in these areas because it's just loose, like it says, loose connective areas where we'll have aggregations of lymphocytes. That's all that is. So instead of them congregating in a lymph node, in the tissue. I'm not going to get into too much about this. We kind of already mentioned these things. And we're definitely going to talk more about the thymus when we get into the next chapter with T cells. But yes, for now, lymph nodes themselves, they say small encapsulated bean-shaped organs. For those of you who have had anatomy, which I think is most of you, maybe you know how big a lymph node is. But the typical lymph node is not like what you think of when you feel like in your neck and you feel the big bumps and whatever like that. The typical lymph nodes and the lymph nodes that they would send us at the transplant lab that we would have to use to extract lymphatic, you know, cells or whatever from lymphocytes, they're about the size of a pea. That's a big one if it's the size of a pea, but usually about half the size of a pea, like a green pea that you would eat. And that's how big lymph nodes are usually. So if you're having an infection and you're able to palpate that, that is going from pea size up to that, you know, palpatable bump. I'm not talking about your tonsils and your adenoids, right? Those are bigger tissue masses. But, and do they play a part in your immune system? Yeah, they do. But they're not just lymph nodes, right? They do other things for the body too. So with the lymph nodes themselves, they're typically really tiny. That's why, like right now, if I touch in my underarm and I know there's like a ton of them in there, I can't feel any lymph nodes. But when I get sick, if I were to get like, I don't know, staph infection, or even when you get like a tattoo, your body can react to that and get some sort of, you know, inflammatory response, and you will feel your lymph nodes swell up then, right? And they don't come out of nowhere. They were just tiny, like half the size of a pea, and now they have swollen up to like what, like the size of a quarter, if not larger. Yeah. So that's how they will work if you guys haven't seen them. Actually pretty tiny, and you can tell if somebody's a smoker. Even if you can't see their lungs, you give me their lymph nodes, I can tell you if they're a smoker. So yeah, anyways, it's gross. They turn black. Right, so major aggregations. I'm not judging, by the way. I am a smoker myself, so I'm just saying that it does show up in your lymphatic system. I'm trying to quit, y'all. All right, major aggregations. You'll have armpit, groin, and neck. We kind of already talked about that. All right, this is what lymph nodes look like. There are layers where you're more likely to find certain kinds of cells. Luckily, you don't need to know about that, but just, you know, so that you can see it and understand. what a lymph node is, that's where your white blood cells are going to hang out and interact with one another if they need to in response to an infection. Okay. Right, so we already know lymph node enlargement is associated with reacting to something foreign that can, if you have them swollen like all over the place or just in a lot of general areas, that could be a systemic illness. But if you have them, like I was saying, like just in your underarm, that's probably a localized infection. So they don't always just all react. Yeah. The spleen. A lot of people know this about the spleen, that the spleen is involved in getting rid of worn out and dead red blood cells. Yeah, filtering out those old red blood cells. Red blood cells, they even have a measure when you do a CBC, when you order a CBC for somebody, there's even a measure of like how old your red blood cells are. They don't call it that, right? But it has something to do with like your mean volume or something like that. I don't know. But it is a measure. You shouldn't have a whole bunch of old ones, but you shouldn't have all new ones either, right? The spleen is kind of what's going to be taking care of that, cleaning out all the old ones that need to come out and, you know, helping your blood stay healthy red blood cells. It's also a lymphoid organ. So it's also going to be filtering out what's in the bloodstream. So this is not going to be dealing with lymph, okay? It's dealing with what's in the bloodstream, and it can filter out what is there in the actual bloodstream. And there are lots of phagocytes, bad guy eating cells, the white blood cells that are going to eat bad things, that are going to be present in the spleen, ready to, you know, react to anything that the spleen might pull out as foreign. Okay. So that's basically what it is. So as it's filtering the blood, it also filters out bad guys, and the cells there can react to it. So we have a lot of macrophages, so we're getting specific with our phagocytes in that particular tissue. Don't worry. All right, so these were our associated lymphoid tissues. We already kind of talked about this. Tonsils do fall into this area. All right, so if you have blood and you send me a tube, if you send me a tiger top tube versus a green top tube, anybody in here ever done phlebotomy? Okay, so I did phlebotomy as a phlebotomist, so for OBI for like 3 years, and then I worked for DLO for like 2 years or something. DLO was a little different. I worked at Southwest Integris as part of like the lab group, but they also do phlebotomy. They're like do both, and their area is weird, but I loved it. I loved doing phlebotomy, interacting with like the patients and talking to them, and they're not super sick a lot of the time in those settings, anyways. And we never went to like beds or anything to get samples, but I liked doing it. And you have all these different colors of tubes, the tops, right? And if you ever go get your blood drawn, you'll notice that too. Each one has some sort of additive in it or not, depending on what the color of the tube is. That's how they mark it, right? So you have a tiger top tube or a red top tube, both of those. They do not have any additive in them at all. The difference between tiger and red is that the tiger has the little wax in the bottom, and it'll separate things out with a barrier. The red doesn't. It just lets your blood sit there. There's nothing in it. Things like a green top tube, a blue top tube, etc., often has something in it to prevent clotting of the blood. All right, so we have that going on. If you let the blood clot in one of those red top or tiger top tubes, it'll clot up. It'll literally, the entire blood turn solid in the tube. You could turn it over and nothing. Okay, that's how solid it should be. It takes a little while and depends on the person and their clotting ability. But sometimes we'll have nurses send up a sample that's a red top tube, and like 5 minutes later they would call and be like, "Where's my results?" I'm like, "You have to wait for it to clot first. Like you have to wait for this to clot." So, but yeah, so it clots, and then you spin it in a centrifuge, and then you'll get the liquid separated from the clotted red blood cells. Okay, the liquid that you get from that, just basic letting blood itself clot and separating it, is serum. Okay, all right, it's different from plasma. If you add some sort of anticoagulant, something to prevent clotting in there of any kind, if it's green top, it's going to be heparin based. Okay. But anyways, if you add something like that in there to prevent the clotting, you can still spin it and separate the red blood cells from the liquid, but they're not going to be clotted, right? If you turn it back over again, they're just going to mix back up again. So the liquid that you get from that is plasma. So it's a different thing. Plasma and serum are different, right? Treated basically, or not treated. Right, but you can do different things with them. That's the important part. Anyways, you guys probably learn more about that. That's not my job to teach you about that part. But anyways, leukocytes are white blood cells. Definitely the same thing. All right, if you hear me call them leukocyte, or you can't hear me call them white blood cells or WBCs, same thing. All right, we can separate white blood cells into a couple of groups. based on what we see in a microscope if we look at them, right? We stain these cells, we look at them. Do they have little granules inside or not? And that's how we're going to separate them, okay? So we have the granulocytes. Well, go figure, the granulocytes have dark staining granules in them, little spots, okay? The granules often represent crap that's in the cells that help fight off the intruders, okay? And then agranulocytes. And that doesn't mean they can't fight an intruder, it just means you can't see it from the stain. So agranulocytes literally just, they don't have them in the stain. No granules in the stain. Agranulum. Okay. They usually have a large nucleus, which is also important for identification. Okay, so we can separate these guys out. The granulocytes, when I ask you about which of the following is a granulocyte, all right, these are the three that I'm going to be mentioning. Now, I will talk about a mast cell in chapter 17, but I don't want you to worry about them as far as whether they're granulocytes or agranulocytes. Okay, we'll talk about them in the context of chapter 17 in allergies. They have to do more with allergies than anything else. And so here, when we're learning about these things, agranulocytes and granulocytes, I'm never going to ask you if a mast cell is a granulocyte or not. Okay, what I will ask you, and I'm not going to put it even on the list, it won't even be a choice. You don't even have to worry about it, right? What I will ask you is which of the following is an agranulocyte or a granulocyte, and I'm going to give you a list. Here's the easiest way to remember the ones that I will give you to choose from, right? If it is a granulocyte, it ends in "phil." Done. Okay. Okay, so we've got the neutrophils. All right, they're the most numerous white blood cell in the bloodstream by far. The basophils, the eosinophils. All right, all of these guys are granulocytes, phils. Okay. The agranulocytes. We can divide this into two groups, the monocytes and its derivatives, and then the lymphocytes. So lymphocytes are agranulocytes. So they are still a white blood cell. They don't have granules. They consist of, the lymphocytes consist of T and B cells. So the letter cells, okay? So they're not got any other name. You don't care about the other ones. There's stuff on here, I realize. You don't need to worry about it. Lucky. So just T and B cells, all right? For the lymphocytes, easy, the letter cells. The other agranulocytes, I already told you that agranulocytes tend to have large nuclei. So monocytes, mono is one large nucleus in there. So those are kind of a giveaway. When a monocyte moves from the bloodstream where it normally is into a tissue and becomes, a worker cell basically in that tissue, then it is a macrophage. It differentiates into a macrophage. The macrophages are by far the heavy lifters of your innate immune system. The macrophages are going to be the ones that are going to be the voice between the innate immune system and the acquired immune system, because we're eating up all of this foreign crap and breaking it down, right? But at what point do we trigger the lymphocytes to come fight? What do they need in order to be told to activate and start, reproducing and all of this stuff? Well, things like monocytes and macrophages, those guys, as they break down whatever they're eating up as foreign, will take parts of it and go and show it to the lymphocytes and the lymph nodes and say, hey, you recognize this, buddy? And when they do, that starts a whole cascade of events that we'll talk about in the next chapter. But so they're important. I would say monocytes and macrophages, super important. I'll talk more about the macrophages than anything because they're already like in the area ready to go, right? Neutrophils, also phagocytes. They just work differently. They have granules. All right, so I mentioned the mononuclear phagocyte system before. It's fancy words. It sounds complicated to me as well. MPS, this mononuclear phagocyte system, is just a bunch of tissue fibers. They call it a reticular system. What that means is that our cells can move in between tissues and organs and stuff like that. This is what they use to move around, okay? They don't just float aimlessly. They don't have like flagella to move themselves around. So they have to have something to move along. And that's what this is, the mononuclear phagocyte system, a passageway between tissues and cells. That's it. All right, monocytes. Monocytes floating in your bloodstream, they're going to become macrophages and dendritic cells. Dendritic cells are just going to be associated with your skin. Okay. So they'll get specialized in whatever tissues a lot of times. In the liver, specialized macrophages are going to be called Kupffer cells. In the lungs, we call them alveolar macrophages because you're alveoli, right? So little tiny sacs in your lungs. oxygen exchange there. That is the preferred location, by the way, inside of those alveolar macrophages, preferred location for tuberculosis to live. It wants to live inside of those, right? So those are important. Skin has these cells called Langerhans cells. Again, those are going to be dendritic cells, very much like macrophages just for the skin. And then the brain have their own sort of macrophage-like cells called microglia, because you cannot have overreaction of an immune response in your central nervous system. Because if you cause damage and inflammation in your central nervous system, that's going to be permanent. There's no recovering from that. So it has its own little system going on there. Yeah, don't worry. We'll talk about the brain later. Talk about that part of it and the blood-brain barrier and all that. So moving on to cytokines. Cyto being cell, and I mean kine. I don't even know. I don't know what that means. Sorry, I don't have one for that one. But anyway, these are usually like signal molecules. Okay, a lot of times proteins, but they're stuff that are exuded by cells to tell other cells what to do. Right. So there's 4 categories of these. The ones that are pro-inflammatory, speaks for itself. The ones that are anti-inflammatory, we're done fighting. We don't need to do this. Let's go ahead and calm down. The reason that your swollen toe doesn't stay swollen, right? Vasodilators and vasoconstrictors to help the blood flow to the area, or to, as you're calming down, maybe even close it off. In fact, whenever you very initially injure yourself, sometimes you'll have vasoconstriction right away, because your body's trying to prevent things like invaders from getting into your bloodstream and moving to other parts of your body. Then we'll vasodilate and bring more blood to the area to bring the cells to the area as well. So it just depends on what's going on at that moment. But anyways, and then we have ones that are growth factors. And these guys are going to help stimulate the lymphocyte growth, right? I keep saying about the B cells and the T cells and how they are the ones that are going to be the heavy artillery. And they're the ones that are really going to be kicking this in the butt whenever we get them going. Well, this helps with that. You send signals to them saying like, no, we need you to come out here. These are examples that you don't need to memorize, but in case you may have heard of interleukins, maybe you've heard of serotonin or histamine, just saying. These types of things, and tumor necrosis factor, or TNF, these things are just molecules in your bloodstream that help signal your cells something to do with the inflammatory response typically, or the immune response. That's all. All righty. So now it wants us to go over physical and chemical defense barriers. All right, we know about our skin. Did you guys know that there is a protein on your skin called keratin that is waterproofing and just helps like keep everything impervious, basically? Yeah. Keratin, protein on your skin, makes it impervious and waterproof. So that usually is one that I test about on the test. So just know what it is. It's a term, not anything crazy, no concepts there. Hair follicles, skin glands, anything to do with your skin as well, like the previous one we were talking about keratin. But the thing about skin also is that you often, if I go like this on my skin, maybe you won't see it right up front, but I'm sure I'm shedding some cells, right? The fact that your skin sheds cells pretty much constantly, it's also shedding any potential bacteria that might be stuck to those cells. So that's a defensive mechanism. This is ridiculous. I feel like this stuff is ridiculous because you don't think of it as being part of the immune system defense. I don't think it is consciously. I think it just works out this way. So I don't know, but hey, there you go. Sweat, mucous membranes, fluids in your eye, the urinary tract, the digestive tract, the respiratory tract, all of that stuff to help flush out microbes. We have in the respiratory tract, we have nasal hairs, right? To catch things that you're breathing in. Copious mucus, yeah, especially this time of year, if you are allergic to things, probably have a lot of mucus in your sinuses and stuff. But we also have mucus down in your lungs, and that's normal. The cilia there, which are little hair-like appendages on your cells, are going to be constantly trying to move the mucus in the area out. And that's so that anything that gets stuck in the mucus in there, we move it out. And then what happens? You cough. You cough, and you cough it out or swallow it or whatever it is, but it's no longer in your lungs where it is going to be more dangerous, right? So that's the idea of the importance of the cilia in your respiratory tree to remove mucus. So when we talk about pertussis later on, pertussis is whooping cough, by the way. We all get our vaccines for it. Most of us should be getting our vaccines for it. What happens is they call it the 100-day cough. And you cough so much, you lose your cilia. Like you just don't have them anymore. There's just so much irritation and coughing and coughing and coughing that you don't have cilia anymore. So even if you get over the pertussis, you're really prone to secondary infections because you don't have the cilia to help move stuff out for a long time. So it's pretty intense. Right, the genitourinary tract, we know about urine, we know about vaginal secretions. Even if we're talking about semen, there are antimicrobial proteins in semen that can help with that too. Our resident microbiota provides microbial antagonism. So taking up the stadium seats so the bad guys can sit down. I know that's my analogy, but that's how I like to think of it. It's an easy one to imagine. So they take up the seats. They also use up the nutrients so that the bad guys can't get hold in the area. Okay, next, in your sebaceous excretions. So whether it's going to be your sweat, your tears, stuff like that, some of these contain something called lysozyme. Okay, it's also going to be found inside of your phagocytes. But anyways, this is an enzyme that hydrolyzes peptidoglycan. What is hydrolyzed peptidoglycan? It breaks it down. This is an enzyme that literally breaks down peptidoglycan. That's all it does. That's its purpose. So sometimes in your tears and your sweat, that'll just be there, ready to kill bad guys, break them down, at least the bacteria, right? We'll also have lactic acid and electrolyte concentration in the sweat that can affect things. In the stomach, hydrochloric acid, digestive juices in the intestines. We already talked about the semen and the vagina. All right, phagocytosis. Let's move on to the cells and what they're going to be doing. The general activities of phagocytes, these are white blood cells that are going to be ingesting, right? So that's what our phagocytosis means. They're literally going to be going around taking up their cell and literally like engulfing things, right? And forming a little vesicle inside of themselves that carries that thing around and whatever, and does stuff with it we're going to talk about. But anyways, these guys that we've been talking about for so long, which cells fall into this category? This is going to be the neutrophils, the monocytes, and the macrophages. Okay, the neutrophils, the monocytes, the macrophages, which remember, macrophages are just differentiated monocytes. Those are our main ones. Okay, so moving on, let's talk about our neutrophils and our eosinophils for a second. Neutrophils, again, they're going to be our phagocytes. They're the most numerous of all the white blood cells in your blood. They are also, therefore, going to be the primary component of pus because there are phagocytes, the primary component white cell in your blood. So they're going to be the ones, hoarding in on these areas where there's foreign stuff. So yeah, they're going to be the primary component of pus. That makes sense. Whether they are dead or active, it doesn't really matter. They're still the most, the primary component there. Eosinophils, if you've ever looked at a CBC or had somebody tell you that you had high eosinophil count, these are usually going to be involved in parasitic infections, typically Hellman's infections. So we don't have those in the United States. And there's a theory we'll talk about in chapter 17 that says that because we are not exposed to, parasites really as much in the United States, that our immune system, like our eosinophils and related aspects of it, aren't modeled correctly because we aren't exposed to those. So now they're overreacting to things inappropriately. So that's the theory. I don't know. They think about it that way. So that's eosinophils. I don't know. Then we have the monocytes and the macrophages, more of our phagocytes. Remember, monocytes turning into macrophages when they go into the tissues, when they're called there by chemical stimuli. So if something is called to somewhere with a chemical stimulus, they get a little molecule that attaches to some sort of surface thing, and they get some protein change and whatever. That's the signal they're receiving. That is typically a cytokine. Right, so we've mentioned those, calling them to a certain area, and it's almost like they will move towards it, and that movement in the direction of whatever signal they're receiving is called chemotaxis. Right, we've learned a little bit about chemotaxis, so that's the same thing here with our phagocytes. They're going to move by chemotaxis. So how's the steps going to go? We have chemotaxis responding to whatever the chemical is in the area. We have ingestion, engulfing the thing that you're responding to, like the bad guy that you've got there. Finally, you see the bad guy, now you're going to engulf it. You form the phagolysosome. So the phagolysosome is two things come together. We have the phagosome, which is the vesicle you made when you ate the thing. And we have the lysosome, which contains, go figure, lysozyme is in there. But we also have those reactive oxygen products we've mentioned previously. Anything in there that we have in our phagocytes to be toxic, to fight something, that's in the lysosome. Think of it just as the vesicle for lysing things, breaking things up. Okay. So what happens is that vesicle, the lysosome, merges with the phagosome, the eating vesicle, and puts them together so that the bad guy now that was in the vesicle is now being exposed to those chemicals. So that's the phagolysosome. Okay. That leads to destruction of that thing. It takes about 30 minutes of exposure to whatever was in the lysosome to kill whatever's in that vesicle. And then we have excretion of whatever products are left over. So that's the phases of phagocytosis and how that's going to work. Okay. All right, so it's going to want us to go over all this stuff, but basically chemotaxis and how we're going to be getting to these areas will respond to cytokines and will also interact with PAMPs. Again, we're bringing this up, but we can recognize, so we get to an area and we recognize, oh yeah, okay, this is the bad thing. I eat this up. So that's via PAMPs. So now when it gets to the area it was called to, it can react via that interaction. So that's the importance of the PAMP in those areas. Remember, pathogen-associated molecular patterns, typically going to be peptidoglycan, LPS, or endotoxin on the gram-negatives, and then the double-stranded RNAs that we see in viruses that we shouldn't have. And then these are just the receptors that I'm not going to ask you about. So we want to be able to react to those PAMPs. Then when they form those phagolysosomes, like I said, it's going to only take about 30 minutes to break down everything in there. It has some oxygen-dependent molecules in there, as well as lactic acid, lysozyme, nitric oxide, and other proteolytic and hydrolytic enzymes, like it says. Just a whole bunch of stuff that can break things down. That's what's in there. Look at it that way. I'm not going to ask you about the details of what's everything that's in there, okay? But I would definitely be aware of that lysozyme is in the lysosome. I feel like that's just kind of self-explanatory. And that we do have those oxygen-based particles in there as well, just because we've mentioned it in the past. All right, moving on to the inflammation. So now we've got our cells there, like our macrophages and whatever, eating up these bad guys and responding appropriately. They're going to be also secreting cytokines into the area. So all of this is going to trigger the inflammatory response, right? It's not just calling the cells there. It's also calling, all sorts of chemicals, all sorts of fluids and everything. So there are there's 5 signs of inflammation, 4 weird words, okay? Latin or something. Rubor starts with an R, redness, okay? Rubor, redness, RR. Let's go down to tumor, actually. So rubor, we'll go this way. Rubor, redness. Next is tumor. We know that tumors are going to look like swellings. If they were actual tumors, we're talking about a tumor. It's like swellings within your tissue, right? So that would make sense that the word tumor would mean swelling to me. Okay. Dolor is the one I can never remember, y'all. So I'll just put a star next to that one, but it's pain, obviously, but like I always have a hard time remembering this one. Yeah, is it? Oh yeah, it's like that makes sense. So okay, so then the last one is calor. Now, to me, rubor, redness, tumor swelling, and calor, heat, they all make sense. So that's how I remember dolor. It's just if you ask me about what the word is for the pain, I'm just gonna like stand there and I'd be like, I don't know. Like I don't know. I just guess that I'm not, I just can't remember it. So anyways, calor. Yeah, does it remind you of calorie? Because it did. So if you guys remember in chemistry, maybe, I know I'm reaching, whenever you burn, like it's like a gram of glucose or sugar or something, whatever energy is given off from that is called 1 calorie, right? A kilocalorie or something like that. So I remember about that being like the heat from burning the thing, or even if you were like working out, would be getting hot over burning calories. So that's how I remember calor. Okay. So if you can remember those 3, maybe the dolor is easier for you. If you speak Spanish, it makes it way easier. I don't. It's a flaw. All right, so our last sign of inflammation that doesn't get its own Latin word is going to be loss of function. All right, you have enough pain and swelling and everything going on there, you're not going to be able to use. Like I said, when I dropped it was not me. When my husband dropped the kayak on my foot, right, and it hurt so bad, like I couldn't really walk on those toes for a little while. Loss of function. That's what we're talking about there. It doesn't have to be permanent or anything. Just in response to the pain and all of that inflammation. All right. Chronic inflammation, when we have all of this building up for a long period of time, can cause damage to the body. Things like your blood vessels, lungs, skin, joints. We see this with autoimmune diseases and long COVID, for example. What is the function of inflammation? Why do we have the inflammation? Number one, We want to mobilize and attract immune components to the area, the cells and other things involved in the immune system, right? So mobilization of the troops. Number 2, set in motion mechanisms to repair damage and localize and clear away harmful substances. So getting our cleaning crew ready now, and our medics in the area. So we're going to need them. And then number 3, destroying microbes and blocking their further invasion. Doing what we can as part of the innate system to achieve that, right? And to activate the acquired immune response, which is going to be not so effective, okay? All right, so anyways, here's how this works. You get your injury in your number 1 area. Cool, we stepped on a nail. That's terrible. You get bacteria, which is the green guys in there. And then there's cells in the area, they're mast cells in this case, don't really worry about it, but they start responding immediately because they're already in the tissue. They can start sending signals to react to those bacteria as well as to call other guys into the area to come help. Typically, those are going to be neutrophils, right? Because there's just a lot of them. So while we're forming a clot with our platelets, so this is number 2 down here. Also, we have that vasoconstriction. Like I said, initially vasoconstriction because you don't want the bad guys getting out of where this happened. Okay, so now as we're moving in neutrophils and stuff into the area, now we can have vasodilation to help movement of all this stuff because we've got our fighters in the area now. Now it's not as risky to bring the blood and open up the bloodstreams, right? So our fighters are there starting to fight the good fight. We're forming clots with our platelets and all of this stuff. As this is happening, we're bringing all this with fluid. So as the neutrophils are brought in, as we're fighting these bacteria, fluid is building up. We've got swelling. It's called edema, right? And then pus is forming. Pus is just going to be whatever is left from the bacteria and the neutrophils fighting, essentially, and the fluid in the area. So it's a mixture of all those things. We know that pus can have active living bacteria in it, but it also has quite a lot of dead bacteria too. It just depends on what phase we're in there. And the body can reabsorb that and interact with that appropriately as it is left to do so. Anyways, after that, we've cleared out all the bad guys. We start fighting off with the lymphocytes that finally arrive to the fight. And now we can heal and form our scar and everything like that. No. Okay, so cool. That's how that works. We don't need to go through all of this, I don't think. But yes, most of it's just going to be signals. We're going to be bringing blood flow and fluids to the area. Exudate is a term referring to fluid that's getting out of the into the extracellular space out of the bloodstream, right? So building up fluid in that area, that's the exudate. And you can even have exudate leaking from the wound as a result of that fluid moving in. So then that's going to be clear or even pus. Then we have edema, which is the swelling and firmness of the tissue from the fluid in there. It's not leaking out, right? All right, how do our cells? I just said to you that we've got all these neutrophils coming into the area from the bloodstreams, right? How do they get from the blood vessel into the tissue? There's not a door that they go through, right? So how do they go from a blood vessel, which we think of as a walled thing, and it is, believe me, into the tissues? Well, the endothelium of our capillaries is very thin, maybe a layer thick of cells. And our neutrophils and other similar cells that will be doing this are capable of changing their shape and squeezing in between cells of the endothelium of the capillaries. So literally just squeezing in. That movement is called diapedesis. That's the word for it. All right, so we know we have chemotaxis to call our white blood cells to the area. Then we have edema, swelling, movement of neutrophils into the area. Then we have attraction of monocytes and macrophages. They're starting to get involved. The activation of lymphocytes and all of this stuff interacting. So B cells, our B lymphocytes, produce antibodies in response to the bad guys. We kind of already mentioned that with our ELISA. The T lymphocytes kill intruders directly, either the intruders themselves or the cells that are infected with the intruders. So we can target them as well. But it takes a while to find the clone that matches up exactly with what you're fighting and activate those guys and multiply them, proliferate them, differentiate them. We have all these different kinds and everything that we'll learn about in the next chapter. So that's what we're waiting on. Once those guys show up to the party, it's done. It's done. So most of you feeling your symptoms of being ill and everything like that, that's your innate response. You're just waiting on the heavy artillery to come in. You're acquired. You're not even going to know. In fact, your acquired is so, like we've mentioned in lab, so effective. Your acquired system is so effective that if you have activated it once and it has developed memory, and that memory has developed healthily, you can walk by somebody who actively has measles. We could bring somebody in here that has measles right now, and we wouldn't even blink an eye. Your immune system will be like, yeah, I got that. No worries. You wouldn't get any symptoms or anything because your immune system works that well. That's your immune system doing that, right? It's not some special shield around you. So cool. I think it's cool and whatever. So fever. We know what a fever is. There's all sorts of ranges that I'm not going to test you on it or anything, but if you're interested, this is what we consider low grade, moderate, and high. Typically, if you go to a doctor with anything below 100, they would roll their eyes at you. If not literally, then in their mind, they would be rolling their eyes at you because to them, 99.5 is not anything to be concerned about. However, you're getting above 100, then you want to consider where that's coming from, right? A pyrogen is anything that causes a fever to occur. Anything. So that can be exogenous, anything from outside of the body, pathogens typically, or their parts. That includes endotoxin, that LPS on the gram negatives, that can trigger fever just on its own. Okay. Then we have endogenous, which is going to be stuff from inside of the body. So that's your cells, that are your immune cells, if they're sending signals out to create a fever. All right. What is the point of a fever? Look at all this, guys. There's a whole slide on what good a fever does, because it does. All right. Inhibits multiplication of the microorganisms. Impedes nutrition for the bacteria. We will remove iron so that the bacteria can't get it. That's what fever will do. It's just part of it. Number 3, increasing metabolism of our own body and our own cells so that we can speed up. Let's say 3A. I don't know, and then 3B, speed up making new cells, which is hematopoiesis, speeding up phagocytosis, speeding up the adaptive immune reactions. He helps speed all that up. And then it increases the ability of those lymphocytes to get to that site where the infection is as well. So all of this stuff happens because of a fever. So when do you decide to suppress versus treat a fever? Slight to moderate fever in an otherwise healthy person, right? You might let it just run its course. I know that when I'm sick and I have a fever that is below 102.5, I think that's my line. I've never had to worry about too much because once you get to 103, you're probably in some sort of real discomfort on top of it, but not that 102 isn't. But if you can manage it, sit through that fever, man, it's doing stuff for you. I promise it is. If it gets too high, take control of it, take some Tylenol. I do not recommend taking ibuprofen. I know it's an anti-inflammatory, but it's bad for your stomach. But if you can alternate, that's probably the better way to go. Yeah. So if you can do that, I would recommend that. It's just not really good for your stomach over time. A lot of people don't know how bad it is. I grew up in a family that took only ibuprofen, and then we took it like it was candy, basically. And then I learned about how bad it is for your stomach, and it is it can be very serious. There's stories of marathon runners who will take ibuprofen for the soreness in their legs when they're running marathons and then be vomiting blood within an hour or something because it's just harsh, causes a lot of inflammation. If your body's already dealing with not wanting to be alive in that moment, then I can imagine it wouldn't like that. But so just be careful with it, okay? It's kind of well known that acetaminophen is safer medication. Taken, of course, according to doctor recommendation. Antimicrobial products, interferon. We've already talked about interferon. It's that neighbor message, right? That you're getting attacked by the burglar. You send a message to your neighbor, and now you have your shotgun ready to go, and you can fight off the bad guy when he shows up. So we can see here, virus shows up. We activate our gene, so we're hiding in our closet. We're calling our buddy. This is the message. This is the phone call. Our buddy gets the phone call and he starts making antiviral stuff. That's the shotgun. Okay. So the virus gets in, we're ready to go. So that's the idea with interferon. We're interfering with the viral infection. You can use interferon to help with fighting diseases by helping activate that whole system as well. So you can provide it to people as a medication. And you can use it to help fight tumors and cancer as well. So it works in a similar fashion. All right, so complement. This is the last of what we'll be talking about for this, no worries. Complement is a whole bunch of proteins and related molecules in your blood, about 50 of them, they're always around. So you don't have to like activate them to like be made or anything like that, which is a good thing. So they're just always ready to go. And then once they start responding to the appropriate things, they just instantly can start dealing with the bacteria. So it's just proteins, right? This isn't cells reacting to anything. This is literally just proteins in your blood that are always there. Okay, it creates a cascade reaction. Once it gets activated, the next thing activates the next thing and activates the next thing and so on. So the first way you can activate it, the classical way, number one, the classical way, the number one normal way that we would learn about is antibodies. Antibodies being around on microbial surfaces will trigger complement. Okay. Number two, the lectin pathway. Lectin is some molecule that binds to sugars on microbes, like stuff we don't have on our cells. Okay. So lectin binds to microbe sugars, activates complement. Okay. The third way, the alternative pathway, we have repeating molecules, kind of like the PAMPs, that complement can interact with that as well, and then trigger this whole pathway. So LPS is the example they give, lipopolysaccharide, endotoxin. All right. So it can start getting triggered by that. So whatever way you react to it, antibody using lectin or by recognizing LPS, okay, you trigger our complement pathway. We have a protein that'll get activated, interacted with, and that's going to allow us to make a cascade products that trigger phagocytosis to help with the phagocytosis. This is something like we get a little protein that comes along and puts a little handle on the outside of that capsule that makes it hard to eat that bacteria. Okay, so it puts a handle on it so that when the phagocyte comes up, it's easier for it to eat it. So that is helpful, right? We've already talked about how difficult it is to eat up anything with a capsule. Now we can put handles on them and start helping eat them. Yes, there's a special word for that we'll learn in the next chapter. All right, inflammation, triggering that whole inflammatory response, that can be started by complement interaction. And then the third thing complement can do, so there's three things, right? Helping phagocytosis, interacting with the inflammatory cycle, and then the membrane attack complexes. This is the coolest thing that complement's ever going to do. So some of these proteins come together and they do so like inside of the membrane of a cell and start building themselves in there, and it creates a hole in the membrane so that crap just can fly out. This is just proteins in your blood that react and do this, just all on their own. They don't need to be having any other signal or anything. They just need to see those things, and when they do, they're triggered and they can start poking holes in bad guys. It's pretty cool. And that's what complement is pretty well known for, is that membrane attack complex. Anyways, it starts out with initiation. Obviously, you see the thing, amplification and cascade, going through all those trigger, you know, turn to these things. Polymerization and the membrane attack. So building up those rings, polymerization and building that up as a whole. So now we can get stuff into the cells as well as leaking out the contents of the cells. So it's useful. That is it. Whew, all right. Wow, that's right on the nose pretty much. Okay, our little question that you can take the picture of for today is going to be, are monocytes important only for the innate immune response, or might they play any other important roles? I did hint at this a little bit in the lecture. We'll talk about it in the next chapter more. But they do have that intermediary role, right, between the innate response and the acquired response. It's extremely important, that role. So talk about that. They're called, believe it or not, antigen presenting cells. So that kind of gives you a taste of what we're going to get into. We don't have to go into the lab. Well, I was going to go over the lab test, though. It's easier if I walk around and remind you guys where the questions were, because then you remember like what it was. Okay, that's fine. We'll just go to do that. It's not like it matters. It's not hot in there, right? Now it's cold. I pretty much bank on it being this way for the rest of the semester. That's how this goes. It's either swelteringly hot or freezing cold in this building.